Hello everyone. This video I'm going to share how to do molecular docking while using AutoDock Vina. So this video I'm demonstrate you how to run a AutoDock, and also one thing I want to say, some people are facing a different kinds of problem when learning of AutoDock. So I thought a very necessary to create in a detail of AutoDock and demonstrate each and every step. So before I'm going to start, I want to share a basic informations about Autodock and a molecular docking. So, what is the molecular docking? So, molecular docking is a study between the protein and the ligand molecular. So, the protein is the largest molecular and the ligand is the smallest molecular. So, questions is that why they required the molecular docking? So, molecular docking is required because they identify the how many active sites are present in our protein molecular, and also they are identify the binding affinity of protein and our ligand molecular. So docking is a two type of docking. First is a blind docking and second one is a site specific docking. So what is the blind docking? So the blind docking, you don't know the active site of protein and you cover the whole protein in a grid box. And now second is a site specific docking. So site specific docking is that the ligand is a bind only the active site of protein, and you know the binding protein of pocket, and you cover the only active site of protein in a grid box, and you don't need to cover the whole protein. Do the molecular docking, we need some software. So first is AutoDock Vina, second MGTools, third Python 5, fourth one is a PyMOL. And AutoDock Vina, MGTools and Pythons, they are generally used in the run of molecular docking study. And fourth one is a Python file. They are used to open the protein and ligand molecular in the PDB format. So these are the four icon, all right. As I told you, I had already downloaded the software, and if you want this software, so you can go in my description box and download this all software. Before I start a molecular docking, I'm going to create a new folder here. So just click in the new and create the new, and create here the new folder, molecular docking. Okay. Now I'm going to keep all unnecessary things like protein, ligand, and other unnecessary file. Now what to need to do it. So go to the program files, and here you can see the MG tools, and now go to the lib file, and here scroll down and click the site packages, and here you can see the Autodock tools. And now here you can see the ID4 bound date files and ID4 parameters date file. Okay, so this file is a which will need to molecular docking. Okay, so just click and paste here folder. Okay, as I told you, here I am create the new folder of molecular docking, and here you can paste here this two file. Okay, now the next need is a go to the program files, and here you can see the script research institute and Autodock, and here you can see these two file Autodock 4 and Autodock 4 and Autogrid 4 files. Okay, so just click and paste here the docking folder. Okay, so these are the four file and which will be need to do the molecular docking study. Now the next we have to require the protein and the ligand molecular. Okay, so how to download the protein and ligand molecular? So in the protein molecular download, we need to go in the website of protein data bank. So here just type PDB and click. Here you can see the first link is a RCSB. Just click, and now here you can tap our the protein name and the download file in the and the download file in a PDB format. Suppose I am here type the dihydrofolate reductase enzyme. Okay, so here you can see the protein molecular, and suppose I need this protein molecular, so just click here, and in the here you can see the right side of download file, and you can see the PDB format. Okay, so just click the PDB format and download this protein molecular. But here I am not download this protein molecular because I had already downloaded this protein molecular in my folder. Okay, so what we need the next, okay, so next need is a ligand molecular. So how to download the ligand molecular? Now to download the ligand molecular, here you can tap the PubChem. Okay, so here you can write the ligand molecular. Suppose here I am writing the trimethyl ligand molecular. Okay, so here we will see the result. Just wait for a few minutes. Okay, so here we can see the this. So here we can see the ligand molecular. Just click here, and here you can see the 2D, 3D, and crystal formats. Okay, but I need only the 3D format, so just click here. Okay, so this is my ligand molecular. Okay, and here you can see the right side the download options. Okay, so just click, and here you can download the 3D conformation, only the SDF file. Okay, so just click and download this ligand molecular. But I already download this ligand molecular. Okay, so I'm not download this file. So you can download here the SDF format. So here you can see the all necessary file to do the molecular docking. Okay, so here you can see the edit for the bounded file, edit for the parameters date file, autodoc for autogrid force, and also the protein molecular in the PDB format, and my ligand molecular is a HDF format. But this ligand molecular I have required in the convert the PDB format file. Okay, because the HDF format are not allowed and not support in the do the molecular docking in a AutoDock Vina. Okay, so we have to need to convert this HDF file in a PDB format. So how to convert this TMP ligand molecular in a HDF file to PDB file? Okay, so we need to PyMOL software. So here you can type the PyMOL. Okay, so this is the PyMOL software, and here you can open. So here you can open the ligand molecular. Okay, so this ligand molecular is a SDF format. Now here you can save this file in a PDB format. Okay, so just click the file and export the structure, export the molecular, and here we can see the option of PDB format. Just click and save. Okay, so here I am save this file in my docking folder. So just click my docking folder and tap here the TMP PDB. Okay, and just save. So now here you can see my ligand is converted in the PDB format. Okay, so here I am just removing this TMP SDF format file. Okay, so we don't need this SDF file. Okay, we just need in the PDB format file. Okay, so these are the all necessary file to do the molecular docking. Now next we need to prepare our the protein and the ligand molecular. Okay, so now you just open the AutoDocs tool. Will be take some time. So this is the AutoDoc tools screen, and where we are going to do the docking. All right. So first thing you need to do it, go in the file and set the preference. So click the file and here the preference and set. Okay. So here you can, and here the startup directory. Okay. So here we can write our the address of locations. So this is my all necessary file, and we are going to need to do the molecular docking. Okay, so here just click and copy this address and paste here the startup directory and make a default and save and dismiss. And now click the file, read the molecular, and here you can see my protein molecular and ligand molecular. Okay, so my protein molecular is a PDB format. So just click and open. So my protein molecular has been loaded in AutoDock Vina. Okay, and also here you can see the red dots of the water molecular. Okay, so and in a left side here you can see the plus icon, so just click and here you can see the chain of A. Okay, so here click the again plus icon and here you can find, so here you can see the different amino acid and also the water molecular and the heteromolecular. Okay, so first we so what the first thing you need to do it, so you go to the edit and delete the water molecular. Okay, so here I am first delete the all the water molecular. Next, okay, now the next step is go to the edit and click the miss and check the missing atom and select the old residue and the dismiss. Now again click the edit format and click the miss and repair the old missing atom. So it will be take some few time. Now click the save as a two sets and the dismiss. Now again you go to the here click the selected yellow dots and removing the all yellow dots. Okay, now click the edit and add the polar hydrogens atom. Okay, so only polar hydrogens atom. Now after adding a polar hydrogen atom, you need to go the edit and click the atom and assign the AD4 type. Now again click the edit and apply the charge of add a Coleman charge. Okay, so in generally the protein molecular it will be apply the Coleman charge. Okay, so just my total Coleman charge is a minus 6.0. So just click. Now my protein molecular has been prepared. Okay, so here go to the file and save and write here the pdbqt. So here you can see the my default folder of molecular docking, and here you can select the all atoms and add and click the OK. Now my protein molecular has been prepared. Now you can go in the folder and check. So here you can see the my protein molecular in a PDBQT format file has been generated. Now what are next we need to do it. So again you click here the ligand molecular and input and open. So here we need to change the format of PDBQT to PDB. Okay, so here click the PDB format So you can see here the ligand of PDB file. So just click and open and display the show the 14 the non-polar hydrogens found and the 10 the aromatic carbon and detected the 7 rotable bonds and also the set the torso also the set the torso to 7. Okay, so just click. Now here you can see the protein molecular and the ligand molecular are loaded in a AutoDock Vina tools. Okay, so what next? So you go to the ligand and click the torsion tree and choose the root, and again you click the ligand torsion tree and detected the roots. Now again click the ligand and click the output and save the PDB file. Okay, so here you just write in a PDB Qt and click the save. Now you can check our the folder my protein molecular and ligand molecular has been generated in a PDB file. Okay. Now next a grid box. Okay. So click the grid box and macromolecular and open. So here my macromolecular is a DHFR PDB. So just click and open. Now again click the grid, set a map type and choose a ligand. So my ligand is a TMP molecular. So just selected ligand molecular. Now next click the grid box and set up grid box. Okay, so here you can see the grid box. Okay, so here I am doing the blind docking. Okay, so here I am covered my whole protein in a grid box. Okay, so you just manually set up a grid box according to cover our your protein molecular. Now my whole protein molecular are covered in a grid box. Okay, so click here the file and choose a close the saving currents and again click the grid to grid box file and output a grid dimension and output grid a dimension file and here write the grid.txt file. Okay, and save here. Now close this grid and now next one is a click here the grid output and save as a GPF file. Okay, go to our molecular docking folder and write here the protein DHFR dot GPF and click the save. Now our GPF file has been generated in our folder. So here you can see my GPF file is generated. Now next one is here click the docking and macromolecular and set a rigid file name. Click here the macromolecular DHFRPDBQT and open. Now again click the docking and ligand and choose ligand molecular is my TMP and click the accept. Now again click the docking, search a parameter and genetic algorithm. Click and accept these all parameters. Now click the docking and output. And here you can see the Lamarckin. Just click and find our docking folder and type here the DPF file dhfr dot dpf. Just click. Now you can check our folder. Your DPF file has been generated. We are prepared the protein and ligand molecular and we need to run in a AutoDock grid. Okay, so click in a run and run in a AutoDock grid. Okay, so here we have to require the auto grid d4 file. So just click the file browser and go to our created docking folder, and here you can see the grid 4 file. So just click and upload here. Now go to the gpf files. So go to the browser, and here you can see our the gpf file. It was already you have to generate it in a this autodoc Vina. So just click and open here. Now you uploaded this gpf file, and now this glg file is a automatic generated. Okay, and now click the launch. So now next click to the run in autodoc, and again you upload here the autodoc 4 files. So just click the browser and click the autodoc 4 files and open here. Now we have to require this dpf file. So go in a browser and click here the dpf file and upload it. Now you upload this dpf file. So dlg file is a automatic generated. Now click the launch. You will now, after done the docking, you will check in our folder. So here the map, you can see the all here generated file, like the map file, glg file, gpf file, dpf file, and also necessary the all generated files in a result. You can see our the created folder. Okay, so f file and dpf file has been generated. Okay, the RMSD table. Okay, so here see the binding energy and cluster RMSD and reference RMSD. So here my binding highest affinity is a minus 6.29. Okay, and also here you can see the dimensional of X, Y, Z of grid box. Okay, so this is the file of DLG file in our result file. Okay, and these extensors are very much necessary how we are prepared the both protein and ligand molecular. Okay, so after you will launch in protein and ligand molecular in a round of autodoc and autodoc grid and autodoc grid and autodoc. So it will be take some time, and after complete our your docking you will see result in our folder. Okay, so many people are facing problem during do the docking because they don't contain the whole folder in a single file. So make sure you save the whole content in a particularly folder, and you will see the accurate result. Okay, so this is the molecular docking study, and also you can save here this image in a PyMOL file. Okay,